Hoje é o dia da defesa do meu mestrado e eu já organizei tudo, tá tudo pronto. A gente já tá aqui na sala de conferência onde eu vou fazer a minha apresentação. Montei o meu slide, tá tudo certo. Me sinto 100% pronta. Só tô esperando da hora da apresentação começar para virar mestre. E eu vou virar mestre em Farmacologia e Toxicologia pela Universidade de Louisville. Tá fazendo um lindo dia lá fora. E olha só quem tá aqui. Olha quem veio me dar uma ajuda, um apoio emocional. E olha que dia lindo.
All right, it's my pleasure today to introduce Nicolli. Nicolli got her undergraduate degree in biomedical sciences from the University of
Yep.
in Brazil in 2021. She then worked 2 years as a clinical laboratory assistant. With her undergraduate work, she had 4 co-author publications. She then moved to the United States and she volunteered in the Mais lab while she was waiting for admission to the PhD program, which obviously she was admitted. She's done very well with her coursework. She bounced around with a couple of different projects in our lab before she finally landed on this one, on the transcriptomic analysis of hexavalent chromium. It's a lot of data that she had to weed through, and she did it all in relatively short time frame, I think. But she's put together a nice story. She's now moving on to Dr. Hardesty's lab. She's been transitioning for the last couple months. She's already got a co-author publication with his group, which is exciting for her. And she also currently serves as the director of travel for the GSC, the Graduate Student Council. I think that's all I have. So, go ahead.
Thank you, Sandy, for the introduction, and thank you all for being here today to listen to my talk. I'm really excited to share the findings that we had with my master's project. So let's dive into it. Okay. So the outline for my presentation, we're going to be talking about lung cancer, hexavalent chromium, the aneuploidy mechanisms that we studied on this project, and then we are going to follow with the methodology and how we sequenced our RNA material, our results and conclusions, and then follow with questions by the end of the presentation. So lung cancer has a high incidence and mortality. So in fact, it's the number 2 cause of cancer in the US. And as you can see on this graph, it's the number 1 cause of cancer death. So the following leading causes of cancer death are colorectal, pancreas, and breast cancers. So if we were to stack these 3 following most common causes, lung cancer will be almost as close as these 3 combined. So this emphasizes how it's important to study lung cancer and to find out more about the carcinogenic mechanisms to be able to mitigate this highly deadly disease. So when we think about lung cancer, it's very common to think about smoking. However, amongst lung cancer patients, 1 in 5 women and then 1 in 12 men never smoked, and most had quit smoking decades before diagnosis. So this raises the question of something else might be going on to this high incidence disease, and this could be metals. So metals are the most common group 1 environmental causes of lung cancer, and group 1 is given by the International Agency for Research on Cancer, IARC, and group 1 means that there is enough evidence of lung cancer induction by these metals in humans. So the most common metals that cause lung cancer are arsenic, beryllium, cadmium, chromium, and nickel. And for the rest of the presentation, we're going to be focusing on chromium. So chromium is a name derived from the Greek word chroma, meaning color. As you can see on this bottom picture, there is a photo of what the chromium metal looks like. And this metal is known for many specific properties, such as hardness and bright colors. As you can see on these top 2 figures, there are a wide varied range of different colors that the chromium salts can get. And in industry, chromium is widely used, and one of those primary uses in industry for chromium is this because chromium is considered a corrosion and oxidation resistant agent. So as you can see on these figures, there is a plane and a boat being coated with a chromium coat to avoid damage and make them last longer. And these are the main industries that still use chromium nowadays. So it's the aerospace and the defense industries. And another major use for chromium in industries is as pigments in dyes, inks, and paints. And as you can see on the bottom picture, there is a leather tanning industry in Morocco, and this became part of the local culture, so this kind of industry still runs nowadays. So exposure to chromium is widespread, so it can enter air, soil, and water. And the main human exposures can be through occupational or environmental exposures. As you can see on the pictures, there is a chromium worker dealing with the transformation of trivalent chromium into hexavalent chromium, and this process is one of the main industries of chromium nowadays. And this process can generate trivalent chromium, which is the non-carcinogenic form, into hexavalent chromium, which is the carcinogenic form. And this process can release fumes and smokes, which can lead to the inhalation of particles of chromium and expose these workers to hexavalent chromium. Also, environmentally, exposures are really common. So any kind of industry waste disposal which is being dumped improperly can contaminate body waters and also the air through air contamination. And this can release particles in the air, so people can expose it to chromium on this way as well. So as I mentioned, hexavalent form is the carcinogenic form, but besides the carcinogenic potential of chromium, it also can cause reproductive and developmental toxicity. However, we're going to be focusing on the carcinogenic potential for chromium. So as I said, chromate is widely used in industries. However, it's a highly reactive compound. So as you can see on this chemical structure, chromate has to be associated with a cation to be stable. And this is really important because the final molecule can be either soluble or insoluble. So whenever we have a sodium or potassium, and these are just the main commonly studied cations for the chromate bound molecules. So whenever we have a sodium or potassium bound to chromate, they form a final molecule of a soluble composition. So they can be soluble in body waters, in any kind of lake or river, and then contaminate the body water. However, if it's chromate, it's bound to zinc or lead, which are again the most common but not the only options. Whenever it's bound to zinc or lead, the final molecule becomes an insoluble molecule. So this is also known as particles, and this is what the chromate workers are exposed to. So we chose to work with zinc chromate for two main reasons. One, it's insoluble, so it's a particle composition, and that's how we think chromium might be affecting directly the lung tissues in vivo. And zinc chromate is also widely used in industries. As you can see, I put up some pictures of different companies that produce zinc chromate primers. So this is still running and widely used in industries nowadays, so people can still get exposed to zinc chromate. So there is some regulation for chromium exposure, and it's given by OSHA, which is the Occupational Safety, Health and Administration, and which establishes an 8-hour time-weighted average permissible exposure limit of 5 micrograms per centimeter per cubic meter. Right? And although it's really good that we have some regulation for chromium, we still need more studies to make sure that this is a safe concentration at any level.
They correspond to either environmental or occupational exposure.
Yeah, so in some of the previous studies published by our lab, they did some of the calculations to normalize how our concentrations relate to the environmental and occupational exposures. So it's not really relative to the occupational exposures, but rather than environmental exposures. Environmental exposures are compatible with the concentrations that we are using.
Thank you. The next question is, you're showing us a lot of transcriptomic data, right? Have you validated any of this in the protein level? And I'm asking because, for example, ORCA B or ORCA A, right? If APC is not working, their protein levels are going to be stabilized. So as a result, the cells might respond by down-regulating their, transcript. So, although you're seeing the transcript go down, because you didn't look at the protein level, now your hypothesis and what's actually happening is not going to match up. So, do you know how your protein and RNA data?
Oh yeah, so with this same RNA material, we already published another works, and the data was validated by qPCR in the gene level. So we have the validation for the whole RNA
No, I'm saying
material.
at the protein level.
And then we also have another projects that were published on the protein level, and they were comparing with the gene level as well. So they are the protein levels match with our DNA material.
Okay, thank you very much.
Vim aqui no mercado agora comprar umas flores para Nicolli e uns presentinhos para ela. Ela ficou lá na universidade, tem mais uma sessão agora só com os professores. Vai durar mais ou menos uma hora e meia de perguntas e respostas sobre a defesa do mestrado dela. Então eu decidi vir aqui no mercado. Nossos amigos lá de Florianópolis pediram também para dar um presente para ela. Então eu tô aqui, vamos escolher umas flores e depois uns chocolates. Qual será que eu pego? Cara, tem um mais bonito que a outra. Da última vez ela levou 2 tipos diferentes e combinou, ficou bem bonito. Vamos ver se eu consigo fazer uma combinação agora de flores pra ela. Acabei de pegar os 2 buquês de flores pra Nicolli e provavelmente eu cheguei em casa e vou tentar combinar. Isso aqui, ó. Aí eu peguei pra combinar essas 2 flores. Essa vermelhinha aqui que eu nunca vi, mas parece tipo uma acerola. E essa flor amarela que tá brotando ainda, então provavelmente ela vai florescer lá em casa. E agora estamos aqui no corredor do chocolate. Problemão agora qual chocolate levar, né? Vamos escolher. Acabei de chegar aqui no corredor de chocolates. Tem diversos chocolates, mas a Nicolli gosta daquela coleção que vem do Ferrero Rocher, que vem aquele Rafaelo, que é tipo um chocolatinho com coco ao redor. Ó, ela ama esse aqui, ó, essa coleção. Indo pra casa agora, acabei de sair do mercado, tentar arrumar o buquê de flores pra Nicolli antes que ela chegue em casa. Antes de ir buscar ela, no caso, no trabalho. Eu acho que ela vai ficar até umas 1h30 por aí, ou 1h da tarde, que é o máximo de 2 horas, eu acho.